There we go. We're started. Thank you, Joseph.
All right. So today I thought I'd talk a little bit about a few different projects that are going on in the lab, just kind of tell you a story about how we use this multidisciplinary approach to a multi-scale approach to design, rationally design nanoelectronics for applications such as sensing and manipulation of biological processes at the nanoscale. So the broad field that we're working is the field of bioelectronics. And the field of bioelectronics is relatively broad, and it's the field of interfacing electronics with biology to be able to measure, manipulate, and sense biological processes. And this encompasses research such as wearable sensors, all the way to implantable sensors, such as this Utah array I'm showing here. Some of you might have seen this. Each of these micron scale pillars can individually record and stimulate neurons when interfaced with the brain. And now the miniaturization of bioelectronics, which is what we do, has two main advantages. First is if our probes are smaller than the size of a single biological unit or cell, we can measure single cells or subcellular activities. And additionally, if our probes are small, we're less likely to perturb the biological system and cause any observer effect. And so the need for this nanoscale electronics for biological sensing, along with advancements in nanotechnology, have given rise to this exciting field of nanobioelectronics in the past few years, which is where most of our work is. And so the way we approach this is a multidisciplinary and multi-scale approach. In my opinion, to understand, to be able to design these nanoelectronics for biological applications, we first have to understand the complex interface between cells and these nanomaterials or the nanoelectronic systems that we're trying to use to sense them. So one subgroup in my lab does this fundamental cellular and subcellular scale studies of studying this nanobio interface. And then we use this information that we get from this nanobio interface to design and optimize our electronics for different applications that I'll talk about today, and to be able to sense and manipulate different biological processes. So subgroup 2 of my lab works on designing these nanobioelectronics based on information that we get from this subgroup 1, which is how the cells interface with these nanomaterials. And then finally, when we design and use these nanoelectronic tools, we develop these large data sets from these biological readouts. And the third subgroup in my lab is focused on trying to interpret these data sets. So understand what's going on and try to answer different fundamental biological questions. So the first subgroup, or the first types of projects that we do, are in this subgroup 1, the nanobio interface. So I'll present one small project that we are working on in this subgroup, which is looking at the cellular responses to nanopillars. Now, researchers have shown that if we look at different parts of the body, the cells, eukaryotic cells in the human body, encounter topographic features in different regions of the body. So for example, if you look at the endothelium basement membrane, it's made up of complex 3D textures in the nanometer range with which the cells are interfaced. If you look at different other tissue, you see that you have these very fine subcellular features that the cells are interfaced with and the cells interact with. And so all these studies suggest that cells have actually evolved to be able to sense nanotopographic features, and the interactions between cells and their environment can regulate primary cellular processes. This is a mechanical cue with which the cells interact, I think very similar to stiffness or viscoelasticity of the substrate, which I'm sure a lot of people in the interfaces program might be familiar with, and a lot of professors in the bioengineering department and other departments at UCSD are working on. Instead of looking at how cells interact with stiffness, this is looking at how cells respond to the shape of the substrate. So one specific topography that we're interested in is these nanometer pillars. So we call them nanopillars. So wherever I use the word nanopillar, it's really just a small scale posts, cylindrical posts, 3D posts that are fabricated on a surface. So we look at the response of cells to these nanopillar structures. So because of the interesting interface between cells and nanopillars, researchers have tried to recapitulate nanotopography in vitro, and including us, who've tried to build these nanopillar structures in vitro and look at how cells interface with them. So one way we actually make these pillars, I thought might be interesting to the audience here, is we use nanofabrication techniques. So right now we're using the techniques we're using the Nano 3 facility to make these structures, but some methods include using, for example, we use a mold and put this mold in an electroplating bath. So we would leave this whole mold inside an electroplating bath, and we would have ions of our metal of interest. So we make these nanopillars out of metals, and we would have ions of the metal of interest in the solution, and we run a current through this whole system, and then the metal gains 2 electrons at this electrode and is able to grow from the bottom up into the mold. So what we end up with is these 3D nanometer scale structures called nanopillars, and we can make them in different shapes and sizes. Interestingly, if we let them grow outside the mold, we can get these what we call mushroom shaped pillars, and they have other applications in biological sensing. So the first very early studies that we and others did was to see whether cells are able to actually grow on these types of surfaces. And we basically just culture cells on, like we would on a petri dish. We culture them on these metallic nanometer scale pillars, and we let them grow for hours, and we can see at least they're dividing. So this is a cell, a scanning electron microscope image of a cell 6 hours after we plate them on these nanopillars, and you can see how after 48 hours they grow, and eventually they can become fully confluent like they would on a regular culture dish. This suggests that they're able to perform their basic biological functions. And us and others have shown that actually the cells can sense these nanopillars. So if you see a cell on a flat surface, it has these random protrusions outside the cell on a flat surface sensing the surface. When you put them on these nanopillars, their philopodia, or these finger-like protrusions, are actually able to extend to each nanopillar and sense the nanopillar. So these are all scanning electron microscope images of cells fixed on top of our nanopillar surfaces, and you can see how the cells are able to interact with them and sense them. And so studies that I won't go into detail showed us that the cells actually interface with nanopillars of different diameters differently. So if you put cells on top of a nanometer pillars, which are with diameters less than 200 nanometers and a spacing of more than 2 microns, the cell actually wraps around these pillars. So it forms small attachments, adhesions on the regions between the pillars, and can actually fully wrap around these pillars. Whereas if you put them on larger pillars, they have enough surface area to attach to right on top of the pillars. So they just are able to spread on top of these pillars. And we also showed that the cells on hollow pillars can actually protrude into these hollow regions. So if you have a cell and you put it on top of these hollow pillar structures, they actually can protrude their membrane into the hollow space. This is, on the left is a scanning electron microscope image of this. On the right is fluorescent labeling, where you can see some cell membrane and F-actin in this case inside the hollow region of our nanopillars. And this is a focused ion beam TM image that also shows a cell membrane can protrude into hollow pillars. So just the summary so far, that some fundamental studies showed that cells can grow and wrap tightly around nanopillar arrays, and they can also protrude into the hollow regions of hollow nanopillars. So why am I talking about all this? Or why is this important, and how can we actually use this knowledge? And so the typical reason a lot of people study these responses to topography, responses to stiffness, is for some fundamental applications such as better understanding disease or using these surfaces for tissue engineering. However, there are some immediate applications to knowing how cells interface with these structures, and that is in our field, which is designing electronics to record from cells. And that brings me to the second part of my talk, which is designing and optimizing these nanoelectronics for sensing biological processes. And we use these nanopillars for higher throughput recording of electrically active cells. So a lot of us are aware that the electrical activity in our brain is really what runs our nervous system, and the communication between our neurons is what allows us to think and solve problems. However, some of us might not completely be aware that the beating of our heart, which is one of our most vital processes, is also coordinated by the electrical activity of our heart muscle cells. So our sensors can record electrical signals from these type of electrogenic cells, so neurons and heart muscle cells. And if you look at the how the electrical communication is performed between neurons and heart muscle cells, you have to know the concept of action potentials. So the electrical communication is performed through these signals called action potentials. So the cell membrane in animals and plants maintains a voltage of around, maintains a voltage between the interior of the cell and the exterior of the cell. This is called the membrane potential. So if you took a voltmeter and recorded the voltage inside a cell compared to somewhere completely far off from the cell, you would see that for neurons, it oscillates. So what you would see, and this is drawn to scale for neurons, if you did this measurement, is that the membrane potential oscillates between somewhere in -90 all the way up to 50 millivolts. And for heart muscle cells, very similar shapes are seen. So these are each called an action potential. And this is how the heart muscle cells and the neurons are able to actually communicate with each other. And so the recording of this action potential is important in a lot of fields of biomedical sciences. And one of the most accepted methods for measuring action potentials is the patch clamp technique. So in this technique, this cell is manually approached by a recording pipette. And then after a mild suction of the membrane ruptures, as shown in this video here. So when the membrane ruptures, the pipette actually obtains access to the inside of the cell, and you can measure the potential inside the cell. So here's the actual experiment here on the right. So the pipette approaches the cell. There's small suctions, there's different methods to do this, but the membrane ruptures, and you can get this beautiful signal which shows the true shape of the action potential. And the true shape of this action potential is important because it has a lot of information about the ion channels opening and closing. And still by the FDA, this is the only technique that is accepted for early drug development to see how drugs are affecting the shape of this action potential. However, there are obvious disadvantages of this technique. So when you do these measurements, the cell dies afterwards because this is an invasive method because of this large pore that is created inside the cell. It's extremely manual, so you'd have to approach the cell under the microscope and kind of patch on it. And so you can imagine how low throughput that would cause this method to be. So there's been some attempts at measuring these electrical signals at a higher throughput. And these are called microelectrode arrays. So these have been around for tens of, I think 20, 30 years, where you can culture a cell on this flat surface that has lots of these small electrodes. So the electrodes are just 2D, and you can have a culture of cells on this surface and record some electrical signals from a culture of cells on top of these microelectrode arrays. And the signal you get is something like this. But the problem is this signal is actually coming from outside the cell. So it, as you can see here, it doesn't show the true shape of the action potential. So it's much higher throughput, but it doesn't include the information that we need. So, but you can see how you can measure 60 channels or even more now with some newer microelectrode arrays. You can measure 60 channels simultaneously on a chip instead of doing one by one using patch clamp. So one idea when these nanopillars were becoming very popular for sensing was that what if we make these really sharp electrodes and they could potentially rupture the cell membrane as done in patch clamp and then access the inside of the cell. However, careful studies have shown that the cell membrane does not actually rupture around these nanopillars. And most of the time, the cell membrane will actually just tightly wrap around this nanopillar, as I showed in the previous studies as well. So the sharp nanopillars are not able to actually puncture the cell and access the inside of the cell. But one discovery was that you can actually use a method that is adopted from the method of molecule delivery using electroporation to actually get access inside the cell. So what this method includes is to apply a very small electric pulse at your electrodes. So you have these nanopillar electrodes and you have a very small electric pulse, very short one, that can create small pores in the membrane of the cell. And now when you are able to apply this small pulse and get these pores, you're able to actually access the interior of the cell. So you're able to do measurements from inside the cell and record the same voltage with some attenuation from inside the cell. So you go from these extracellular signals, which have a very low information and very low signal to noise. With electroporation, you can get some shape of the action potential. And so for making these pillars to be able to record from these electrical signals, when we first started working on this project, we were trying to develop an easy fabrication technique to make these nanopillar electrodes. So we came up with the kind of same method that was used before I showed you, which is called the electroplating technique. This is a bottom-up technique, so you can grow the pillars from the bottom up inside this mold. And this method is very versatile. You can fabricate electrodes of various metals and different shapes and sizes. And so we're able to make all these different shapes of electrodes for these sensors. And so we were successfully able to record intracellular electrical signals using this method. So we have our nanopillars, and when we apply a small pulse, we can get these intracellular signals. Sorry, just give me 1 second to plug in my laptop because it's dying. Give me 1 second.
looks like your laptop might have died before she had a chance to plug it in.
Yeah, I think so.
Sorry about that. My laptop did die, but now I'm back. Can you hear me?
Yep,
Yes,
sounds good.
Yeah. So where I left off, we were able to measure very still high, low signal to noise, but you can see clearly how the shape of the action potential changes. So we can measure these extracellular signals, and then if you apply a pulse, you can get these intracellular signals using these nanopillars. However, we did some careful studies to see why we were not getting great signals. And one was that the forces that are exerted by our heart muscle cells, which is what we were recording in this case, actually can detach our nanopillars from our surfaces. So when we did some SEM images of our sensors after the culture of these heart muscle cells on top of it, you can see that the cells don't particularly treat the nanopillars with care. And so this is also some SEM images of the cells with the nanopillars, and you can see the forces that are exerted by the cell can completely detach these nanopillars from the surfaces. So we realized we have to come up with a better technique to fabricate these sensors. And so you can imagine all these forces from the cells that are beating on top of our sensors can actually deform our sensors. And so we came up with this other approach, which is instead of a bottom-up approach, which is growing the nanopillars from the bottom up, we thought we can actually use a top-down approach where we etch these pillars down into the surface. And so now the bottom of this pillar is continuous with the substrate, and it's much more robust mechanically. And so we can coat a small layer of metal on top of these, and then that could be our electrodes. So we were actually able to get a 10 times better signal with these new nanopillars. So again, we have our extracellular signal with that much information. We can apply a small electric pulse and get these nice intracellular signals. So now we can get signals in the millivolt range with a very successful rate of electroporation. And then finally, we thought if we could even get a better attachment of the cells with our electrode, the better seal between the electrodes and our cells. And this takes me back to what we talked about in the fundamental studies, is about how cells are able to protrude into the openings of hollow nanopillars. And so we thought whether we could use this idea to actually get a stronger signal from the cell by increasing the attachment between the cell and the nanopillars. And so we thought of this new shape for electrodes, which is because the cell protrudes into the pillar, but not all the way, whether we could make these new structures called nanocrowns or semi-hollow nanopillars, where the cell membrane would protrude into the hollow opening and actually form more attachments with the base of our electrode. And so with this, you can get this very tight seal between the cell and the electrode. And so it was very easy to make these semi-hollow or nanocrown electrodes. So if we have our original nanopillars, we can actually just etch the top of these nanopillars and then etch a little bit into this base of the nanopillar. And so we can come up with these very fine nanocrown electrodes with which the cell can interface. And we call them nanocrown just because of the shape of the top of the pillars. And so we were able to get our highest and most prolonged signals with these new shape of electrodes. And you can see here, this is the extracellular signal. This is our intracellular recording after a small electroporation pulse. And we can get signals up to 40 millivolts with patch clamp. You're measuring about 100 millivolts. So we're getting very close to the patch clamp amplitudes because of this strong seal between the cell and the nanocrown electrodes. And so these electrodes allow prolonged intracellular recording. So this is one example of a cell recorded. So at the first time point, we have extracellular signals. We apply this pulse and we get this, in this case, 63 millivolt amplitude recordings. And over time, we can measure this at even up to 1.5 hours, measuring the same cell. And the cool thing about this is that this pore that we create in the cell does seal over time. So we can measure the same cell over multiple days without damaging the cell. So here's one example of a recording from the same exact electrode with the same cell over multiple days. So as you can see, even if I measure the first day and I apply a small pulse and get an intracellular signal, the next day I come back, the cell is back to this extracellular signal, suggesting that the membrane pores have completely recovered for the next day. And you can repeatedly porate the cell over multiple days and do this electrical recording. And also then we can do this recording over multiple channels. So this is a recording showing up to 58 channels, recording intracellular action potentials over 60 channels simultaneously. And so one of the potential impacts is for, again, as I mentioned before, the FDA still requires this test to see any drug that is going to be on the market, whether it, test it for cardiotoxicity. So how it affects this action potential is one thing that the FDA is interested in for any drug. So one way to quantify that is look at this action potential and measure what is called an action potential duration. Really what it is just the width of the action potential at different time points. So for example, APD50 is this width that I'm showing here. And I won't go into detail about it too much, but really it's just this width to quantify the shape of this action potential. So it's very common to, for example, have a plot that shows the drug dose on one axis and this action potential duration on the other axis. So with patch clamp, you would be able to measure a few cells and create this plot. But now that we have our method, we can actually do this at a much higher throughput. So this is one, for example, recording from our devices. And you can see how we can add different doses of the drug over time and look at the shape of the action potential and its change for the exact same cell. And so, for example, here, we tried multiple drugs that have different effects on these shapes. And again, these shapes are dependent on how the drug interacts with the ion channels. And so it's not, so it gives a lot of information about how that is happening. So for example, a high-risk drug like dofetilide, we can see for the same cell as you add the dose, the higher doses of the drug, it prolongs this action potential. And now for one single drug, we could do this on multiple cells, on multiple simultaneously. And we can also do this repeatedly on different experiments without, and we can wash the drug out and repeat it. So you can get several data points in a very limited amount of time. So it makes this experiment much higher throughput. So it could accelerate the discovery of drugs at the very early stages. And then finally, this brings me to the last couple minutes to talk about this final subgroup that we have in the lab, which makes it actually very multidisciplinary and multi-scale, is to interpret the results that we get from these high throughput data sets. So when we have these large data sets, we really need to use more advanced data analysis tools to be able to interpret the results. And this brings me to a story that I was training for my first marathon, and I joined this running club in Palo Alto, and I met this Dr. Annalisa Palowski. She's working at Google. So she's a head of a lab at Google, and they were actually interested in pharmacology as well. And they were looking at higher throughput methods to record signals from neurons and from heart muscle cells. And so she invited me actually for a talk in their group to present this work that we had done. And I did that. And then Google had these small grant opportunities. So we wrote a grant together for that opportunity, and we were able to get that grant to work on some machine learning projects regarding the data sets that we obtained from these nano electrodes that we designed. And I will just touch on one small project that we're working on, and that is whether we could use our platforms to actually obtain intracellular information from extracellular signals. So as I mentioned, one of the advantages of our platform is that we can record extracellular signals and intracellular signals from the same cell at the same time. So imagine that on the left, I have my electrode that is sitting outside the cell. So I measure these extracellular signals. And on the right, I have my after my electroporation, I can record intracellular signals. So we can actually record, and we have recorded, thousands of pairs of extracellular and intracellular signals. So the question is whether we could actually obtain this intracellular information without having to do this intracellular recording, and by only doing extracellular recording. And the advantage of that is still extracellular recording is easier to do, less invasive, and in vitro, that's the only method that's possible. So we are working on some projects here, which is trying to use convolutional neural nets to actually predict and reconstruct these intracellular signals merely from the extracellular inputs. And we've had some success. Again, in the interest of time, I won't go into detail, but we're able to predict pretty accurately the intracellular signal simply from the extracellular input. And this we've only been able to do in cardiomyocytes so far. So one project that we're working on is to make our model more generalizable, understand the features of the action potential between the extra and intracellular that actually allow us to reconstruct the intracellular signals. And we're collaborating with Professor Rose Yu, which I think would be a great addition to this program as well, to actually use physics-guided AI to understand this relationship between intracellular and extracellular action potentials. And then finally, we want to try to do this for other cell types, such as neurons. And just to summarize my talk, the key takeaway points from the different projects that we did was that cells can grow and wrap tightly around nanopillar arrays, and the cell membrane can actually protrude into the openings of hollow nanopillars. And we can use this to actually obtain a tight coupling between cells and nanopillars and design very sensitive nanoelectrodes that can record these electrical signals from these heart muscle cells. And then finally, these nanoelectrodes can have a potential to accelerate drug discovery at a higher, by doing pharmacology at a higher throughput. And then the final thing that I might have conveyed is that exercise is good for your career, because the way that I was able to collaborate with Google and get some money from Google for the project was actually through running and meeting some people through running. And with that, I'd just like to acknowledge some of the people that were involved in the project that I presented here. I worked with several chemists and chemical engineers, mechanical engineers, data scientists, and bioengineers and biologists, and some very talented teachers that were visiting the lab when we were doing this project, which we had a lot of fun working with, and we were able to come up with curriculum for their classes to take back and teach based on the research that we were doing. And with that, I'd like to thank you and answer any questions that you may have.
Thanks for fantastic. Obviously, your lab and your research and your collaborations are beautifully matched to the interfaces program. Because we're running a little short of time, I think we might want to wait till the end for any questions so that Andrew can do his presentation. But thank you again very much for a really a really great talk and great introduction to your lab.
Sure,
It's
thank
beautiful
you.
work.
Thank you.
So I guess I'll go ahead and share my screen. So hopefully you can all see that.
Take it
Looks
away,
good.
Andrew. I'll save time on introductions. I think probably
Okay.
everyone knows about it anyway.
Sure. Okay. So hi everyone, I hope you're doing well. So today I'll be talking about my research in developing a predictive model of circadian plasticity. And so this talk is a bit of an extensive background on sleep and plasticity because these are huge areas of research kind of separate with each other, but hopefully by the end of this talk, I can convince you that sleep does in some way affect synaptic plasticity. So I first want to talk about what sleep is, which is a bit of a silly question because we sort of all kind of know what sleep is. One definition I kind of like is something that graduate students probably don't get enough of, but for the sake of this talk and sleep researchers out there, I'll define it a little bit more rigorously. And to do this, I'll use an aid of a electroencephalogram, or more commonly known as EEG, which are essentially measures of brainwave activity. So usually this first measurement, they measure brainwave activity when you're awake, but drowsy. And so when you start to fall asleep, it'll measure your brain activity during sleep. And when you're first dreaming, which is this stage called NREM sleep, which is non-rapid eye movement. And they also measure brain activities when you're in deep sleep in this stage called REM sleep, or rapid eye movement. And basically what you observe is like a pretty large difference in brain wave activities between these stages of sleep. So a little bit more strictly speaking, sleep is defined as this state between unconsciousness and consciousness when you're digesting information from that day. So it's a bit nice that we understand sleep a little bit loosely speaking, but let's get down to some specifics. So interestingly enough, you can model the sleep-wake cycle as a two-process model. And this is more or less visualized in this graph, where in the red, we have this process called process S, which is essentially also commonly known as adenosine buildup. And we also have something called process C, which is basically a measure of the circadian rhythm profile throughout the day of most organisms, which is more commonly traced using melatonin, but that's a little bit too specific. But simply put, the sleep-wake cycle is governed by this accumulation of adenosine during the day and decrease of adenosine during nighttime. So when you sleep. So using this information, we can look to some molecular machinery to explain this graph in a little bit more detail. So essentially what this little diagram is saying is that during the day, your body uses up ATP, or energy, and produces its derivatives ADP, AMP, and eventually adenosine as a byproduct. So this in turn kind of explains this accumulation that we see of adenosine throughout the day, since more and more adenosine is being produced because a lot more ATP is being used throughout the day. And so when we have more, and this is probably a little bit more intuitive, but if we have more adenosine in kind of the extracellular space, these adenosines are a little more likely to bind to their respective receptors, A1 and A2A, which are essentially these sleep promoting receptors. So thinking about it, if these are the agonists for these receptors, then when these adenosine bind to these receptors, these neurons will kind of emit these sleep promoting signals. So as more adenosine bind to these receptors, we see this increase in sleep pressure and will be more likely to sleep. So talked a little bit about the molecular machinery behind adenosine in sleep. But like I mentioned, the title of this talk, I kind of want to connect it to synaptic plasticity. So we'll first define what synaptic plasticity is. So thinking about it, we know that plasticity is basically the brain's activity to adapt to change to new information. So therefore, synaptic plasticity refers to the change that occurs at these synapses, which are these junctions between the neurons that allow them to communicate. So if you put some stimulus in your system or synapse that increases this calcium ion signal throughput, you see an increase in this synaptic strength, resulting in a change in synaptic plasticity. So there are many well-known calcium signal pathways that result in synaptic plasticity, such as the glutamate-mediated calcium signal pathway and NMDR-induced pathways. But however, during literature research, there's been more and more emerging evidence that adenosine has some involvement within these pathways, which is highly suggestive of adenosine's role in modulating synaptic plasticity. And so taking a step back, we want to know how does sleep relate to synaptic plasticity? And so there's many hypotheses, and one in particular that's very popular is the synaptic homeostasis hypothesis, which proposes that the fundamental function of sleep is essentially this restoration of synaptic homeostasis, which is challenged by this synaptic strengthening triggered by learning during wake and during development. So that's a lot of jargon, but in other words, sleep is basically the price we pay for plasticity. So an increase in synaptic strength has various costs at the cellular system level, including higher energy consumption and greater demand for the delivery of these cellular supplies to the synapses, which leads to cellular stress associated changes in support cells, such as glial cells. And increased synaptic strength also reduces the selectivity of these neuronal responses, and essentially it saturates the ability to learn. So by renormalizing these synaptic strengths, sleep reduces the burden of plasticity in other neurons and other cells while restoring this neuronal selectivity and ability to learn, which leads to this consolidation of integration and memories. So essentially sleep leads to learning and memory. And so what the basis of my project is that I want to investigate what is this underlying molecular machinery that ties sleep to neuroplasticity. And from the previous slides, we kind of get some bits and pieces on what this black box actually is. And so we'll hypothesize that this black box can be adenosine. And we hypothesize that, adenosine coupled with these previously mentioned calcium ion signaling pathways, that the intervention of adenosine in these coupled pathways can be the right link to link circadian rhythms and how they affect synaptic plasticity. And so from my literature search, we've determined that in many folks works in our own lab that calcium ion presence leads to synaptic plasticity, and looking at these pathways that involve these glutamate and NMDR receptor pathways is sufficient enough, because you have an increased calcium ion activity in the presence of these ligands and receptors. So it's essentially sufficient to say that these pathways lead to synaptic plasticity or synaptic activity. But so what I propose is that what happens if you couple these receptors and integrate their signals. So instead of having all these separate pathways, you kind of want to couple them and look at the interactions between all these pathways and receptors and ligands. And so basically the schematic is based on 3 models that I've come across in my literature search. And the first one that will focus is on this cyclic AMP and pKa pathway. And so this is based on the Ohadi model in 2018, which Dr. Jahed is a previous postdoc in our lab. And so she built a mathematical model of calcium-induced cyclic AMP and pKa pathways to integrate basically different steps in the modulations of signaling components in this pathway. So this model was constrained using experimental data in the literature, and the model predicts that the components in this pathway serve as a frequency modulator of calcium dynamics, such that this pathway only picks up the lower frequencies of calcium influx. So in other words, that means that this coupling of cyclic AMP and pKa is a leaky integrator of these calcium dynamics, which essentially this work had shed light on kinetic mechanisms for robustness of the cyclic AMP and pKa responses in dendritic spines. And so experimental measurements of these calcium oscillations in dendritic spines also show that calcium oscillates spontaneously, and these measurements are used to generate predictions of cyclic AMP and pKa kinetics for different calcium outputs. So essentially, this coupling of cyclic AMP and pKa are important for looking at spontaneous calcium oscillations. So we have the schematic again, but now we'll look at a different piece of the model. And so we'll look at this glutamate mediated calcium signal pathway that involves a PLC, or known as a phospholipase C, which hydrolyzes to generates this IP3 pathway, creating this positive feedback loop for the intercellular calcium. And so another model that we looked at is this Colisel and Bell model, which is previously derived from the cooling model, which was developed in 2008. And essentially they're looking at these cardiomyocytes and modeling transient IP3 in cardiomyocytes. But the difference between that and this model is that they actually have inclusion of the endoplasmic reticulum. So it's a little bit more of a complete pathway, essentially creating that positive feedback loop for between IP3 and calcium. And lastly, I want to talk about the last piece of the model that I have currently, and that is involving these VGCs, or voltage-gated calcium channels, and how the presence of these channels actually modulate calcium, or intercellular calcium. And so, this work done by Zhang et al. In 2019 essentially is a model of it essentially built a model describing the spontaneous oscillations of calcium in astrocytes. So a little bit different for now, but not completely different. But what is of interest is that we built this model to observe how these spontaneous oscillations rise in the presence of different types of voltage-gated calcium channels. So it's on the right here. And it's just what they find is that in the presence of these voltage-gated calcium channels, you observe these spontaneous oscillations if certain channels are blocked. You kind of see the absence of these oscillations. So this is a little bit indicative of stating that these spontaneous oscillations arise due to these interactions between these voltage-gated calcium channels and this IP3 and PLC pathway. And so in my next slide, interesting enough, you'll see that kind of just from this simple parameter sweep of the resting membrane potential, that there's essentially a regime where you would see these spontaneous calcium oscillations for a given range of the resting membrane potential. And so basically these 3 models provide a general framework of a ODE compartmental model that I can certainly piece together to create a neuron cell-specific model that's reflective of the schematic that I've shown in the previous slides, minus the adenosine input. And so what the main result in my model is that we see this present. So now that we have this presence of glutamate, which is essentially the only difference between the coupling of my model and the previous models, is that it's in the presence of glutamate, you kind of see more pronounced calcium oscillations, which is what you would probably expect to see, since the Zeng model doesn't have this presence of glutamate, and we know that glutamate is in some way modulates calcium activity. So this is kind of what we expect. So it's not something extremely novel, but it's something that you would expect if you put this into additionally into the model. So so that's the gist of what I've done so far. And so the next part is adding this adenosine input, which is a bit challenging because there are, at least to my knowledge, no existing models that have adenosine and kind of these like coupled calcium signal pathways. And there is not a whole lot of data describing the interactions between adenosine, calcium, and all these voltage-gated calcium channels. So the next step is to start thinking about how to actually put that in the model. So moving forward, once we consider adenosine into the model, we kind of have to ask the question, how is adenosine then regulated? And so from that proposed model, so definitely you can, with the proposed model, we can see kind of the role adenosine has in kind of modulating these calcium oscillations and calcium activity and in turn synaptic plasticity. But it certainly does not inform us of anything to do with the circadian rhythm, since there's no kind of additional adenosine coming into the system. And so we kind of had to look back at the schematic we mentioned before with ATP breaking down into adenosine. So at some point we might have to consider putting in extracellular ATP to kind of capture the molecular machinery involving this kind of circadian behavior of extracellular adenosine. And so I come to my last slide. And so the main takeaways are that, I've basically established a framework where I can then add adenosine in the system and see how this external adenosine affects these calcium pathways of interest. And something worth mentioning is that although I plan to build a neuron cell-specific model, a lot of the intermediate models or even the final model can be applied to probably other cells. So probably other cells of interest are, one that comes to mind is skeletal muscle. So we can kind of ask the question, how does changes in sleep or sleep deprivation affect calcium activity in skeletal muscle? And so I kind of mentioned this before. I kind of want to extend it to include these interactions between the voltage-gated calcium channels and adenosine and its receptors, and then further extend this model to include the circadian behavior adenosine related to sleep. So we'll be talking about timescales of hours to days. And so essentially this will be a multi-scale modeling effort, which is definitely new territory for me. So it's a bit challenging, but yeah, so sorry for going through that so quick, but yeah, that's basically the end of my talk. So I'd be happy to take any questions.
Thanks, Andrew. That was great. Does anyone have any questions for Andrew? Or for Professor Zainab? Jahed?
I did actually have a question for Dr. Jahed. I was curious about different cell shapes and how you might kind of adjust accordingly for a cardiomyocyte versus a neuron if you change your nanoelectrodes and kind of that approach.
Yeah, that's a very good question. And that interface study is what's really important because, for example, the membrane properties, the size of the cell, everything changes when we try to record from neurons. So that's what we actually are having difficulty with right now when we're trying to modify our sensors to interface with neurons. So the size, I think, of the electrode, I would say is very important. And because the most important thing is the adhesion between the cell and the electrode. So we're trying to change the size of our electrodes and make them a little shorter to see if we can get the neurons to wrap around them as well.
Okay, interesting. Yeah, that makes sense. You figure, obviously the dendrites are much different than a kind of flatter
Yes, yes.
cardiomyocyte. So.
And we do denser arrays for neurons because we want to be able to like measure from multiple points on a neuron, whereas the cardiomyocytes, we are measuring from one location on it.
Okay, yeah, that makes a lot of sense. Okay, yeah, that was really interesting. Thanks so much.
Thank you. Thanks, Andrew, for the great talk. Sorry for rushing you through your talk a little bit because I didn't plan for exactly 20 minutes.
No, it's totally fine.
Yes, great job, Andrew.
Thank you.
And also a great example of another really interesting multi-scale problem, especially in time.
Exactly.
Yeah.
Any more questions?
Yeah,
Well, if
hopefully we
not,
can do
let's-
this in person next time with more interactions and
Yes, yeah. Sorry about the change
that's
plans, but
not a problem.
hopefully Kelly will be recovered soon.
Hope so. Hope you feel better soon, Kelly. Sorry for what happened.
I'm sure I will. Like I said, ice and ibuprofen, amazing combination.
Yeah.
Okay, everyone. Well, we'll see you next time. And actually, I guess there won't be a next time. Well, will there be one more before the end of the quarter?
No, well, we're going to have one more, but it's just going to be with the trainees and their mentors.
Right.
So that's the last one for this quarter, but we'll be starting back up in the fall. So
That's right.
looking forward to that.
Yeah, we look forward to seeing everyone and your mentors for the last session, lunch session of the year. So, thanks again to Andrew and Professor Jahed for their great
Yay,
talk.
thank you.
Thank you. Thanks, everyone. Bye.
Thank you,
Bye-bye.
everybody. Bye-bye.